I guess the fact I said there wouldn't be clicker questions means that people decided they had better things to do this morning. And yes, I am trying to record this as we go. There is a exam here at 10:15, so I'll try to be good about finishing actually as close to 10 as possible. Those of you who have not filled out your online course evaluations, please do so ASAP. Your chance to say what you liked or didn't like about me and the course and so on and so forth. I will not see them, don't worry, until after all the exams, which is when is the exam? At 8. Tuesday at 8, exactly. Be here. So that is it. What are we going to have on the exam? Everything since we had on the last exam, and nothing particularly more than that, so not comprehensive. Lots of people have asked me about this. So we started out talking about translation in this section of the course. I do not expect you to remember the genetic code. I expect you to remember one thing about the genetic code. When you have an A, U, and a G, what do you have? You have a start codon or a methionine. Start codon if it's the beginning, and methionine if it's somewhere in the middle of your protein. Important things as far as the genetic code are concerned. It's degenerate. We've got 64 codons, 3 of which are stop, which is way more than the 20 amino acids that are generally used. So the genetic code is degenerate, which means you can have multiple codons that code for a single amino acid. Nice example would just be leucine here, for instance, 6 different codons that code for 1 different amino acid, but then also similar kinds of amino acids, i.e. their chemical structures, are also together. So leucine, phenylalanine, isoleucine, and valine are all very similar in terms of their side chain hydrophobicities. So having a change to change the particular amino acid under those conditions is not likely to be a big change in terms of your protein. Again, could be, but generally is not going to be the case. And again, as usual, stop me as I'm going through here, and we can talk about it a little bit more. There seems to be a considerable amount of confusion about open reading frames and reading frames here. So the take-home message with this is really all about the genetic code. So genetic code is a triplet code, and it's non-overlapping. So where you start is absolutely critical in terms of what protein you actually get out at the end. So any individual stretch of nucleotides has 3 reading frames in it. And so reading is just thinking about the ribosome. So ribosome goes 3 nucleotides, 3 nucleotides, 3 nucleotides, 3 nucleotides. And so in this case, there are 3 reading frames, and I think I may have misspoken about +1, +2, etc. It's just there are 3 reading frames. An open reading frame is where you have a start and then some stretch of amino acids before you get to a stop. And that's all that your open reading frame is. This reading frame down at the bottom, the 3rd reading frame, could be part of an open reading frame if somewhere over on this side off of the screen is an AUG in that frame of every 3 nucleotides. And somewhere over here there's always going to be a stop, so that's not going to be an issue. So is this clearer? Yeah, in the back.
If there are multiple ribosomes on one strand of RNA, can one ribosome read it differently from another one, or do they all start at the same place and stop at the same place?
Okay, so that's a great question, and it really depends on where you start. And so that's why translational initiation, which is just about what we're going to get to in like 2 slides, is so important. And the where you start can make a huge difference in terms of what one you get back. It's almost always going to be the same start, however. There are very rarely cases, take virology, we'll talk about some really cool ones, that you actually have ribosomes reading in different frames. But it's, yeah, again, it's great because that emphasizes that where you start is absolutely critical in terms of what reading frame you use. Okay. How that reading frame gets turned into protein, i.e. how the genetic code is translated, is not the ribosome. It's really the amino-acyl tRNA synthetases, because the amino-acyl tRNA synthetase that recognizes the tRNA, recognizes the anticodon loop down here, and puts the amino acid onto the 3' end of the tRNA over here. The amino-acyl tRNA synthetases have both the activating the amino-acylation activity here on each of the amino acids and this ligation activity to the tRNA. And again, it's all about the specificity of the amino-acyl tRNA, which determines, amino-acyl tRNA synthetase, I should say, that determines what amino acid gets put on here. The ribosome has no clue. The ribosome is just going to put in whatever tRNA that has the codon-anticodon pairing appropriately. And that gets to one of the other, I think, confusing parts for some people, is the whole idea of mischarging. This is a description of the experiment that showed that. This is the experiment that showed that the ribosome is clueless. It just takes whatever codon-anticodon pairing which is happening and will incorporate that amino acid that's attached to that tRNA. And so that's, that's it for our tRNAs, et cetera. How do you get translation to take place? It's the ribosome. Ribosomes have 3 positions on them: the E site, the P site, and the A site. So the exit site, the peptidyl transferase site, and the amino-acyl site. How you get translational initiation: you have to get the start codon and the initiator tRNA, which is a different tRNA than the one you'd use in the rest of elongation. These guys have to associate with each other. Of course, it's different in bacteria and in eukaryotes. Once these have associated, then you can have the large subunit of the ribosome that associates. The reason this only has to happen after this is that there's no space in the middle of the ribosome between the large and small subunits to get that tRNA into the P site. So once those are together, then you can have elongation, which is where you have the new tRNA comes into the A site. You have transpeptidation that takes place. Then you have the ribosome that translocates. You get a new tRNA, et cetera, until you get to a stop codon, strangely written over here. We then have a release factor that associates with these codons that don't have tRNAs that match them. And that leads to dissociation of the ribosome. So we'll step through those 3 steps. And again, thanks for the question there about where things start. For bacterial ribosomes, it's all about the 16S ribosomal RNA and a sequence in that 16S ribosomal RNA that is complementary to a sequence in your messenger RNA. This is, again, completely different from what goes on in eukaryotic systems. That's your ribosome binding site or the Shine-Delgarno sequence. That's next to wherever your start codon is. And so here shown as AUG. And I'm sure all of you remember, because you've been studying, that some bacterial ribosomes can start at GUG or UUG. Well, how can they do that? They do that because these things are right next to really good ribosome binding sites. So it's the proximity to this ribosome binding site that says this is the AUG that you will use. And this is why it's very rare you end up in a different frame, because you've got that very strong specificity in bacteria. Okay, so that's how you get the small subunit of the ribosome to the messenger RNA and select your start codon in bacterial systems. Eukaryotic systems are different. Here you have the, sorry.
The Shine-Delgarno sequence, is it the mRNA or the rRNA?
The Shine-Delgarno sequence and the ribosome binding site are all in the messenger RNA, so in the mRNA. And so the complement is going to be what's in the 16S. Got other questions on this? Sorry, if I'm like on a roll, stop me, yell. So eukaryotes are different, but particularly in terms of their messenger RNAs. Messenger RNAs are being made in the nucleus, they're getting exported, and then once they've been exported, they have these other proteins that are associated with them, particularly the end of your RNA, which has a 5' cap structure, that interacts with EIF4E, which displaces the cap binding complex. So we talked about the cap binding complex way back when for the last midterm. And that is what gets put on to help that messenger RNA get out into the cytoplasm. Once it's there, it gets exchanged for eukaryotic initiation factor 4E. 4E and 4G associate with each other, and 4G is this real bridge between the cap and the poly A tail, but not the poly A tail, but the poly A binding protein, which are these green ovals right here. So any eukaryotic messenger RNA in the cytoplasm is present in this loop structure. And this loop structure is what's going to serve as the template to get translation to happen. So how does that happen? You have this structure, which then will associate with the small subunit of the ribosome together with your initiator tRNA. And so all of these guys come together, bind to the 5' end. But the 5' end is not where the start codon is. So you have to have this complex translocate through this 5' untranslated region, UTR. And if you've got structures, you've got proteins that are bind there, that will clearly stop any kind of translational initiation because you're not allowing the ribosome to move through that to get to the start codon. Once it does get to the start codon, you have base pairing between your initiator tRNA, the start codon, and then hydrolysis of GTP by eukaryotic initiation factor 2. There's an IF2 in bacteria, very, very similar. And basically all this is doing is checking to make sure that the base pair interaction is correct. You know, AUG is pairing with the appropriate anticodon again on that initiator tRNA. So once that's happened, again, that's been released. You have the large subunit that comes together. Now you have translational elongation. Translational elongation is practically identical between bacteria, archaea, eukarya, any cellular organism, and the proteins have different names, but they really have very, very similar functions. So as you've started, now you'll have your P site tRNA that has a growing polypeptide chain attached to it. The next aminoacyl tRNA comes into the A site. If it has the correct match, then EFTU, or eukaryotic elongation factor 1, hydrolyzes GTP. This tRNA is released, and peptide bond is formed, catalyzed by the ribosome between this amino acid here in the P site and the amino acid which is on the A site. Now you actually will have your polypeptide chain attached to the tRNA which is in the A site. Then you have translocation. Translocation moves this tRNA which is in the A site into the P site, and you go through this whole process again and again and again. It's, again, GTP hydrolysis. GTP hydrolysis is the protein that's associated with the tRNA. Conformational change happens only when you have the appropriate base pairing interactions, which then releases the tRNA. If that tRNA is not bound properly, it will dissociate. And this is probably really the really slow step in translation, is going through all of the tRNAs to find the appropriate one, which is actually going to match the codon which is present in the A site. If you get to a codon that doesn't have an appropriate tRNA that can bind to it, these are your stop codons, then you have release factors, which basically pretend to be a amino-acylated tRNA. The ribosome tries to make a peptide bond to the amino acid which isn't there, and that gives you your termination. This peptide is lost, and then the release factors, together actually even with the elongation factors, cause the whole ribosome to come apart, large subunit, small subunit, etc. These now can recycle, and either, if you're talking about a bacterial system, find the next ribosome binding site, or if you're eukaryotic, your 5' end is right next to the 3' end where you just had termination, so it's not far to go to find the cap to continue to go round again. So this is the process of translation. Questions on translation at this point? Okay, briefly wanted to talk about some of the weirdness in translation. A number of people have asked me about this as well. How do you get frame shifting, selenocysteine addition, etc.? So both of these, both selenocysteine incorporation and frame shifting, have to do with secondary structures that form in the messenger RNA. So secondary structures, just base pairing interactions that are going to form in the RNA. Those base pairing interactions give you double-stranded RNA, and then you have some kind of protein that associates with them. In the case of selenocysteine, that protein now brings a selenocysteine tRNA to the ribosome where you would otherwise have a stop codon. Selenocysteine, once it's in the A position of the ribosome, because it's not a release factor anymore, it's now a real tRNA, the ribosome is perfectly happy to make a peptide bond here, and then this selenocystyl tRNA will be translocated into the P site, and you can continue. This is now, again, it's completely dependent on a structure which forms in your RNA, and that structure is going to be dependent on sequence. You can't get a structure unless this can actually base pair. So it's only going to happen where you have an appropriate sequence in your messenger RNA that's not coding for protein. This is coding for a secondary structure which is going to lead to the incorporation of selenocysteine. The other form, of changes in translation that we've talked about here, there are other modifications as well, but these are sort of the two big ones. Frame shifting. Frame shifting is also, kind of getting back to your question before about if you start at one position, all those ribosomes staying in frame. Yes, unless there's a frame shift which takes place. And these frame shifts are also going to be dependent, on some kind of secondary structure in the RNA. But this now seems to be independent of protein. So it's just a secondary structure in the RNA. And the secondary structure in the RNA causes the ribosome, when it translocates, to not translocate properly. And so it's almost always in a stretch of nucleotides which are identical. Not always uracils, but identical nucleotides. And the ribosome, probably what happens is it runs into this loop structure and then backs up one nucleotide and then tries to run through it again. Most of the time here, it turns out that it reads through it perfectly fine. 90% of the time in these viral RNAs and most of the other ones, you get regular translation in this frame. 10% of the time, you have a change in frame, and you end up with this, in this case, a much longer protein. But it's just a change in the reading frame. And this is one of the very few examples where you have ribosomes on one RNA that are in different reading frames. But they're not going to be all running in different reading frames at the same time. They will have shifted their reading frame. And you can have reading frames that go forward, some that go backward, etc. But again, all dependent on having some kind of RNA structure. So also means it's sequence dependent because you've got to have some kind of sequence in order to form these secondary structures. Once we've made our peptides, Are
secondary structures in RNA relatively common or rare?
Are secondary structures in RNA common or rare? Turns out that secondary structures in RNA are incredibly common because if you have a sequence which can base pair to itself, you're going to form secondary structures. The key is that these are going to be specific secondary structures and relatively stable secondary structures. And so in the case of selenocysteine, it's actually something which is a binding site now for a protein. And then in these cases for the frame shifts, they're going to be quite specific secondary structures. But if you look at RNA inside a cell, we always draw, look at these nice RNA here all stretched out in one little, or one piece, one nice long line. That's not the way that RNAs are. They're all bound up with each other. Okay, other questions on translation. So once we've translated, we've got a polypeptide. That polypeptide in and of itself, when it comes off the ribosome, is not active because it's just primary sequence. It's just a stretch of amino acids. So that has to come together to its final three-dimensional structure, and in many cases also has to associate with different proteins in order to have its final activity. So this process, going from a linear polypeptide chain, or slightly wiggly as they have it here, to its final structure, this is what's called protein folding. And I have the unique conformation. I was going back and looking at these last night, so like I didn't really talk too much about this. But if you have a, excuse me, the linear amino acid sequence of a protein, 99.99% of the time, that sequence is going to form one structure. Now, it's that 0.1% of the time, which is the interesting thing, so that's why everything I have in quotes. But it really is that primary sequence which determines the structure. And how you go from primary sequence to structure is still a very open question that people are trying pretty hard to figure out exactly how that happens. Because it's really easy to figure out what sequences are. It's a lot harder to figure out what structures are. So if you could predict your structure from sequence, that would be a massive breakthrough. Part of that has to do with the fact that these 20 amino acids can interact with each other in a whole bunch of different ways. And sometimes those are the wrong ways as far as the function of the protein, and that's where the chaperones come in, is to stop the wrong kinds of interactions. And so here we have our nice example, again, an unstructured protein which forms a little bit of structure, the molten globule. This is a little bit of secondary structure, I think probably most of the secondary structure, which, as we've talked about before, is all about hydrogen bonds forming between backbones. So each of the peptides, and doesn't matter what the side chain is, can form some kind of secondary structure. Tertiary structure is where those side chains are coming together. So most of the secondary structure, not much of the tertiary structure, and then through an iterative process, together with some of these chaperones, you eventually end up with the right protein. Most of the time, if you don't end up with the right protein, there's a couple of ways to deal with it. The proteasome, which is this protein garbage disposal inside the cell, which is driven by ubiquitination. So misfolded protein, ubiquitin ligase will put ubiquitin on it, and then that ubiquitin will have more ubiquitins added to it. That's the signal to send it to the proteasome and have it be degraded. And in a few cases, you have protein aggregates, which are bad, and this can cause a number of problems. But fortunately, this is a very thin arrow compared to all of these other arrows in the process. So now I've got our proteins. Great, wonderful. So let's talk about how you get those proteins in the first place. We talked a little bit about cloning, and I have cloning here in quotes because this is somatic cell nuclear cloning. Most of the time when you talk to a molecular biologist about cloning, they're talking about cloning a specific small piece of DNA. In this kind of cloning, the Dolly cloning, we're talking about cloning whole organisms. So very different kind of thing, and unfortunately it kind of gets squished together. The way that the somatic cell nuclear cloning, I think is actually a great name for a change for some of these things. So somatic cell nucleus, you actually get the nucleus from a somatic cell, and you put it into an enucleated egg. And that nucleus from the somatic cell can lead to production of a complete organism. And that complete organism is going to be a complete molecular clone of this organism and the somatic cells that came from it in the first place. Yeah?
Shouldn't there be an extra arrow from epithelial cells to making it to a somatic cell?
So question is, shouldn't there be an extra arrow from epithelial cells to somatic cells? Epithelial cells are somatic cells. So there's somatic cell versus a germline cell, splitting those two up. And so these are, and a germline cell, that's what you use normally for fertilization. So that's where you would be normally getting this from. But here, in this case, it's something which is differentiated and would normally not be a germline cell. Yeah, differentiation between germline and somatic there. Yeah? and then just segregate to.
I was confused because how does an egg just develop into an embryo? Don't you need something else? You need a sperm or something to get an egg to a zygote?
Yeah. So in this case, you actually then have your diploid. So the question is, don't you need a fertilized egg in this case? And it's amazing, but it does work with the non-fertilized egg. So basically, for those of us who are males, we're just contributing the DNA. That's it. So if you bring in 2 copies of the DNA from the somatic cell, that's plenty. No, don't need those stupid men anyway. Yeah.
What are the issues that arise with the lack of mitochondria when you get the nucleus in the somatic cell?
Okay, so what? Yeah, because these stupid men don't contribute the mitochondria. So now the question is there, what are the issues with, and in fact, this is not a complete clone. I'm paraphrasing here your question, because there is actually still some genetic material which is left, for want of a better word, in the egg, and those are the mitochondria. And the mitochondria are actually then all derived from this egg cell. So yes, it's a clone in terms of the nucleus. It's not a clone in terms of the mitochondria. In fact, it's a clone of the mother's mitochondria, which is true for all of us. We've descended from our maternal mitochondrial lines. There are exceptions. Actually, a really cool paper that was published, I think, earlier this year. It turns out there actually are a few mitochondria you can get from your father in a few rare cases. But for the most part, yeah, it's just the mitochondria is going to be from the female. And it turns out that you can. There's a group, in fact, at OHSU. Sorry, it's getting a little aside here, but Shukrov Metolipov, I think, probably mispronouncing his name, has actually been looking a lot at mitochondrial diseases. And in fact, there's now some cases of where you can get mitochondria from one donor, but then the nuclear DNA from 2 other donors. So actually a triparental, as he's talking about, which actually seems to work really well for solving some of these mitochondrial diseases. And in fact, there are kids in England who have been born with the mitochondria from someone who's not their mother. Amazing stuff. Really kind of cool and mind-blowing, but kind of a aside, rather than this. But great question. So yes, it's all about the DNA in the nucleus, plus the mitochondria and whatever else happens to be in the cell. And then one other thing you could always ask about here is what about the other proteins that are present in the egg? So if you had some particular protein that had a activity, say a transcriptional activator, for instance, that wouldn't be in one organism but is in the other one, you can think about some of the genetic memory and epigenetic markers which might still be present in the maternal egg as well. So yeah, it's all about the DNA and some other stuff. But as far as we're concerned, it's all about the DNA. So that's what we're going to talk about. DNA, and particularly DNA binding proteins. DNA binding proteins, this is how you get that regulation, how the DNA that is in an egg eventually turns into a cow or a whole organism. And that's mostly having to do with the gene regulatory proteins interacting with the DNA. The vast majority of gene regulatory proteins interacting with DNA are going to interact in the major groove of the DNA, and in the major groove of DNA, because that's where you get sequence specificity. That's how you can have, binding by a specific protein to specific sequence. And that's how you can regulate the genes which are next to it. What the amino acids are that interact, we wish we knew. We're actually not very good at doing this, and this is why people are really excited about CRISPR, because CRISPR-Cas, you can use an RNA which will hybridize with the DNA, and you get much better sequence specificity, and you can actually design it that way. We talked a little bit about dimeric DNA binding proteins. The key with the dimer is you now have one protein, so that's the green, Nike swoosh, I guess, here, binding to DNA. Each of these binds to the same sequence, but they're in opposite orientations. So you have a protein that binds to 5 nucleotides here, 5 nucleotides here, ends up actually giving you the specificity binding to 10 nucleotides in a particular position. And then if you have these dimers, you can mix and match them as well. So almost all sequence-specific DNA binding proteins, be they bacterial, be they archaeal, be they eukaryotic, are going to bind as dimers. How they dimerize? Lots of different ways, but a couple of the really common ones, particularly in eukaryotic systems, are leucine zippers and helix loop helix. These are dimerization motifs. They're not DNA binding proteins. So leucine zipper, everything which is above the DNA here. The helix loop helix, everything which is above the DNA here. They bind to DNA through these basic helices that are present in the major groove of DNA. Basic helix present in the major groove of DNA. You can totally have a leucine zipper that has nothing to do with the DNA binding protein. You can have a helix loop helix that has nothing to do with the DNA binding protein. We can get, sorry.
What's like the structural differences between helix loop helix and helix turn helix?
Okay, structural difference between helix turn helix and helix loop helix. So helix turn helix is a DNA binding motif that you find in bacterial proteins. The helix loop helix is a dimerization motif that you find in eukaryotic proteins.
So DNA binding motifs can't dimerize together, but these can.
Yeah, and so they're serving completely different roles here. Sorry, so the question was, helix turn helix. Paraphrase. So helix turn helix motifs can't dimerize with each other, and the answer is no, they can't. And it makes kind of a lot of sense because if you think about what the proteins are that are, the amino acids that are not binding specifically in the major groove, what are those going to be? They're going to be positively charged. What happens if you try and bring two positively charged proteins together? It's not going to work very well. So you don't have the helix turn helixes binding to each other. They almost always have separate dimerization motifs somewhere else in the protein. And then helix loop helix then really do interact with each other.
So are zinc fingers also in eukaryotes, or are they?
Zinc fingers are almost used exclusively in eukaryotes. And then the question again that was on the exam in the one biology class I took as an undergraduate, can the zinc finger get caught at a leucine zipper? You can think about that. There's no particularly correct answer to it, but okay, sorry. Was that answer your question? Okay. So I mentioned again, dimers are really useful because you can have this higher specificity. You can have fewer proteins that will still bind to DNA in various different places. But the other big advantage of dimers is you can get cooperative binding. And cooperative binding just literally as far as you have a protein increase in protein concentration. If you have a dimerization and that dimer binds really well to DNA, but the individual monomers do not, you can have much more of a switch-like DNA binding versus concentration, a sigmoidal curve, as opposed to this exponential increase curve. Sorry, this is a asymptotic increase curve. So that's really three reasons that dimers are very good. One, you get higher specificity. Two, you can get heterodimerization. And three, you can get cooperative binding. Okay, wanted to briefly talk about methods because everybody gets confused about methods. Methods are cool. The way that we know pretty much everything that we've talked about in the rest of the class, like why you get this kind of cooperative binding. So electrophoretic mobility shift assays, almost everybody calls these band shift assays, are where you have a DNA, say like a promoter, and you're interested in finding proteins that interact with it. And those proteins that interact with it will cause it to go through a gel more slowly because each of these is bigger. And usually this is just one protein. I don't like this example because it's got a whole bunch of different ones here. But an example of this is now you have, and this is almost always going to be a nuclear extract or some mixture of a whole bunch of different proteins that you put onto a column, and then you see the proteins, the total protein concentration off of this column. Each of the fractions you get off of this, so go straight down here, it'd be fraction 1, 2, 3, 4, 5, 6, 7, 8, 9. Here you don't have any DNA binding. In fraction 10, you have something that binds to DNA. Fraction 11, you have something that binds to DNA. Fraction 12, you have two things. 13, you have 3 or 4 things, etc. And so each of these fractions, now you can say, okay, I've got certain numbers of proteins in this one, and they all bind to DNA. So it's a way of seeing that you've got DNA binding to a particular piece of DNA. Yeah?
Do you do the chromatography first or the gel first?
Okay, you have to do the chromatography first. The question is chromatography first or gel or the band shift first? Chromatography first, because you're looking at each of the fractions that are coming off of the column. And you're basically looking to see, and again, I don't like this example because there's a whole bunch of different proteins. So you probably should find one that just has one. And so you've got all these different proteins that are coming off of your column. You want to know which one binds to your DNA. And you look through all of the different fractions on the column and you find, okay, whatever is in fraction 23, that's the one that I'm looking for, as opposed to all of the other ones. Okay, this just gives you binding to a particular stretch of DNA. If you want to know the actual sequence that your protein binds to, that's when you do your, I say precise sequence, I should say. That's when you do DNA footprinting. And DNA footprinting is you have a specific piece of DNA. It can actually be exactly the same sequence that you had for your band shift experiment. But now you chop up this piece of DNA into smaller pieces. And if you chop up this piece of DNA into smaller pieces, if you don't have any of the protein around, say from fraction 23 off of the column, then it looks like this. You've got all of these DNAs. You separated them on high-resolution gels. Each of these corresponds to one nucleotide. Then you do exactly the same experiment with protein around, and it will stop the DNA from getting chopped up between this length and that length. And so you'll be missing all of these pieces. And so that footprint right here says that your protein binds to these nucleotides right here. So again, it's just a way of getting very specific sequence that your protein has interacted with. This is great and wonderful, but who wants to do single proteins and single pieces of DNA now? We're all in genomics. We're in omics world, so we're really interested in where proteins are interacting with DNA in a cellular context and usually multiple different places inside the cell. We talked about chromatin immunoprecipitation, and then the recording went bleh, and I didn't really have a good chance to talk about this cut and run method. But basically what this is doing is very, very similar to the chromatin immunoprecipitation followed by sequencing, only the way that you get your smaller piece of DNA is much different. And so here what you do is you bring in an antibody, and that antibody to your favorite protein also has a nuclease attached to it. And that then, the nuclease will cut the DNA right next to your favorite protein and chops just that small piece out of your genome. And then you can sequence all of the DNA that you have in the genome, particularly these small pieces. And all of these small pieces you can then line up relative to your genome down here. So this is a genome that, fortunately, the Human Genome Sequence has determined, Human Genome Sequencing Project. And then the height of each of these bars is how many sequences you have found in that particular position. And here we have an antibody to lysine 27 methylated in histone H3, and you find that they're bound here in the areas next to where you have lots of genes being transcribed. So it's just another way of looking at where your protein has bound to DNA, but now you're looking at it in the context of a whole cell. Okay, questions about any of these methods things or the method stuff that I didn't talk about? Okay, let's talk about regulation. The lac operon is a great way to review regulation in bacteria because it's got both kinds, positive regulation and negative regulation, even though positive regulation doesn't like exist. The negative regulation, which was what was originally found, is the lac repressor. And the lac repressor binds to the lac operon in the absence of lactose and blocks the polymerase from transcribing this gene. Unfortunately, or fortunately, unfortunately for Jacob Monod, turns out there's also positive regulation, and you actually need a mixture of positive and negative regulation to get the appropriate regulation of the lac operon in E. coli. And that positive regulator is CAP or CRP. positive regulator binds to DNA in the presence of cyclic AMP, which says you have low glucose. Low glucose means you want to stimulate the binding of an RNA polymerase, but you only want to stimulate it in the absence, or say the presence of lactose. Presence of lactose, the repressor comes off, and the absence of glucose, the activator comes on. Now you have expression. So repressor binds to DNA, blocks RNA polymerase, activator binds to DNA, stimulates RNA polymerase. Usually RNA polymerase binding, but in the case of NTRC, actually it's activation of transcription by an already bound RNA polymerase, and that acts at a distance very similar to what happens for eukaryotic transcriptional regulation. So eukaryotic transcriptional regulation, a much more complicated process, A, because we have much bigger genomes in eukaryotes, and B, because we also have chromatin and nucleosomes associated with the DNA in eukaryotic systems. So a whole combination of different proteins, but for the most part, they're still basically doing the same thing, bringing the RNA polymerase and all the other general transcription factors that it needs to a promoter. That's giving you the activation of transcription, and blocking that from happening is going to be repressing transcription. So the processes here, they can be functioning a lot like you have in bacterial systems, right next to where you have the assembly of the pre-initiation complex, the promoter. But very often these are long distances away. They can be upstream of the promoter, they can be downstream of the promoter, and all of these guys interact with either the general transcription machinery itself or this mediator complex in between. And this can be positive, this can be negative, etc. One of the other much more, I don't know what the right adjective for this is, complicating factors in terms of eukaryotic transcriptional regulation is not all of the proteins that are going to regulate transcription interact directly with the DNA. Many of them interact indirectly with the DNA. And so those are our co-activators or co-repressors. They will bind to DNA, but only through other proteins. So if you've got DNA binding proteins like this, they in and of themselves don't serve as a gene regulatory protein. They only serve as a gene regulatory protein together with some kind of co-regulator. In this case, a co-activator. This co-activator could interact again with components of the general transcription machinery, but a lot of the co-activators, and for that matter co-repressors, are really involved in chromatin regulation. So a co-activator could be a histone acetyltransferase, puts histones, puts acyl groups on histones, which leads to the chromatin falling apart or association of your pre-initiation complex. A co-repressor would be involved in methylating some of the histones, and methylation of histones then leads to compaction of the DNA, no access to the promoter by the general transcription machinery. So the fact that you've got these things that are all working either through chromatin or through space and looping means you actually have to have some way of making sure that the sequences next to a promoter, and next to a promoter I can mean tens of thousands away, an enhancer can be tens of thousands of nucleotides away, only works on that particular promoter and doesn't work on all of the other 20,000 promoters that are present in any particular genome. So there's got to be a way that a gene is somehow insulated from the rest of the chromosome. How does that work? Insulator elements, and again, element, whenever you see element, that's going to be a nucleotide sequence, and you'll have the factor which associates with the element. Factors are all proteins, elements are always going to be parts of your nucleic acid. So here we have our insulator elements, The insulator elements are there to make sure that this green sequence here, which is where you have your binding site for all your transcriptional regulators, only is going to regulate gene B. It's not going to regulate gene A or C or D or E or F or any of the other ones. How that works is that we have these insulator elements that are bound to proteins, and that makes a loop, and that loop seems to separate this regulatory sequence from this gene and all other genes, so it only works on the gene which is right next to it. How exactly that works and why it works in terms of through space is still an interesting question. A lot of it just does seem to be, yes, regulators can work through space, but they work a lot better on something that they're nearer to. And probably a lot of it really is this proximity thing. Okay, so talked a lot about chromatin so far. Chromatin can actually be something which is modulated in terms of getting transcriptional activation, and also can be modulated in terms of getting transcriptional repression. I'm just talking about activation here, but again, repression is going to be a very similar one. So often we'll have some kind of histone modifying enzyme. I already talked about this before. These are our histone acetyltransferases, our histone methyltransferases. These can modify histones next to where you have your gene regulatory protein. Again, activator or repressor doesn't really matter here. These can bring in histone chaperones. Turns out that the histone chaperones are actually not as common as you have nucleosome remodeling complexes or chromatin remodeling complexes. These will move the nucleosomes along the DNA in such a way to either block the Tata box, where you can't then get pre-initiation complex to associate, or open it up so you can have the pre-initiation complex that associates with it. And so chromatin remodeling complex, again, moving stuff back and forth. Had a couple of questions in office hours about what the heck is a bromodomain and what the heck is a chromodomain. So the bromodomain is a domain. It's part of a protein, folds up in a particular structure. That structure is, of course, structures have functions. The function of a bromodomain is to bind to acetylated histones. So if you have a protein with a bromodomain, it will associate with acetylated histones. And so here we have the example of one of the proteins in TF2D has a bromodomain. It's going to associate with this acetylated histone right here. Also, you'll have bromodomains that are going to be associated with histone read-writer complexes, because that's what they're doing. They're reading a acetylated histone. So you get an acetylated histone, you have a protein that's got a bromodomain that's associated with it. The flip side of the chromodomains, where you have binding to methylated histones, and that can lead to gene repression, all kinds of other things. And also, we didn't talk about this too much, but we talked about DNA methylation that happens in gene silencing. Those DNA methylases have chromodomains that are associated with them, so they'll associate with the methylated histones, but then they will also then methylate the DNA. So protein-protein interactions, really key for all of these processes. One of the things that seems to confuse more people than anything else I talk about in the whole term is this even-skipped gene regulation. And no, I'm not going to ask a specific question about this on the exam. But the idea is, unless I give you a lot more detail than, say, what was in one of these clicker questions, the idea here is that we're talking about a combinatorial control. So combinatorial control, we've got repressors, we've got activators, and it's the presence or absence of those different activators or repressors. And in this case, it's in a particular position in the embryo, but it could be in a particular cell, in a particular cell type, all kinds of different situations. But the basic idea here is we're looking for the combinations of these proteins that lead to gene expression. And this is just for the stripe 2. There are the 6 other stripes of even skip that you have in the embryo. We're not concerned about them here. They've got different proteins that are associated with them. The big deal here is that just these 4 regulators, giant, which are the green blobs here, they should be blue, but they're green, and then cripple, which is the other repressor here in these little red hats, and then the 2 activators, bicoid in yellow, which is in fact in yellow, and hunchback, which is in blue down here. The only time you're going to get gene expression is wherever you have just the activators bound and none of the repressors bound. So the only place that this happens in the whole embryo of these 4 proteins is right here. The 2 activators are here. Over here, you've got the repressors. They're going to block the binding of activators. Over here, you don't have the activators, and you're blocking the binding of the activators. So again, the message here is not, I want you to totally understand gene expression in Drosophila. I don't think anybody totally understands gene expression in Drosophila, with the possible exception of Christine Nusslein-Volhard. But the idea is thinking about these kinds of structures and how you end up getting gene regulation. Yeah?
So the clicker question for this one?
Yeah.
It was, you're missing giant,
Mm-hmm.
and then you'll get expression anterior to stripe 2.
Correct.
Positional expression.
Mm-hmm.
But anterior to stripe 2, I assume would be like stripe 1, and I thought these proteins only regulate stripe 2, and the other stripes are regulated by maybe these, but more as well?
Okay, so that's a great question. And so, again, clicker question said anterior to stripe 2 if you are lacking giant. So maybe I didn't make this clear here. You have activation of this gene in this particular region, but you also have repression of this gene right outside of that region due to the presence of these activators. And so that's the only thing which is regulating the fact that you're not getting expression right next to it is the presence of these repressors. So if you get rid of the repressor, and that's sort of what I tried to show here. Sorry about these little X's, but the X here, so the X over the bicoid here and the X over the hunchback here, that's because you have giant here. And so if you didn't have giant, then these guys would all be bound just like you would have it here.
So stripe 1 has...
Okay, stripe 1 is way the heck over here.
Okay, so you're
Just talking about right here in stripe 2, because that's the only thing that was asked about.
So is this like within the stripe?
Well, no, next to the stripe. So there's the stripe, but the stripe is so it's expressed there and only there. And the regulatory protein is actually, it's that regulatory region is not just for expressing in the stripes. Also, it's for not being expressed where it's, because that gene is present all throughout the whole embryo. Okay, yes, no? Yeah. But if I were to ask a question like the clicker question, I would give you basically all the data on this slide. It's a waste of paper. I shouldn't do it. Okay. I'll think about it. Have I written questions for this lecture yet? I'm not sure if I have. Yeah, we're a little behind here, but that's okay. So did want to just briefly talk about yeast mating type switching. No, I don't expect you to remember what alpha 1 and A do, but that you do have this process of cleavage at the mat locus and exchange through homologous recombination, which gives you A's versus alphas, and it's the expression of the proteins present here, alpha genes and or A genes, that lead to the activities that you'll expect from a alpha cell or an A cell or a diploid cell. And that these guys here, the extra copies that you have in your genome of those regulatory proteins, they're silent because they're in parts of the gene which are in heterochromatin. So heterochromatin shutting down genes, the mat locus is active, and you can get the changes that happen there. How it happens, again, it's activators and repressors. The details here are not that critical. Again, I'm going to speed up a little bit here, so slow me down if there's stuff that you want me to spend more time on. So idea of the genetic switch, again, The actual proteins that are involved here are not critical, but the idea is you have feedback loops. So you have one state that is decided because you have an activator which is activating its own gene. That gives you a positive feedback loop. So you have one choice and you stay with that choice even after lots of rounds of cell division. CpG islands, again, common subject of discussion in office hours. CpG islands happen because methylation happens. Methylation is going to happen at CpGs. Over evolutionary timescales, those CpGs are going to turn into TGs if they've been methylated, because that's the standard DNA damage. If there's some part that is selected so you don't have methylation of those CGs, then they'll be maintained in genetic time. They'll be selected for. And what that means is that you've selected for CGs in regions where you're not going to be methylated. You're not methylated. The DNA is not methylated in regions which are going to be constantly active, i.e. in promoter regions. And so that's why you have CpG islands. It's because they haven't been methylated. And there's reasons then that they've not been methylated. So we talked briefly about DNA imprinting. DNA imprinting is just the methylation of some sequences in the DNA. And I actually just read a paper yesterday that said imprinting is probably way less frequent than anybody thought it was. So why we even have it in the textbook is another interesting question. But the idea here is, again, if you've got methylation of a particular sequence, then you change what proteins are going to be associated with that sequence. And so here we have an insulator element. This insulator element normally is going to function if we have binding to that. It'll block activation across this insulator. However, if it's methylated, you don't get binding. The insulator doesn't work. Sex determination in flies. Somebody asked me a question about this also on the emails. Is it really just the presence of double sex and which form it is that determines the sex of the fly? Yes, it really is. Flies have very bizarre sex determination systems. And so if you don't have double sex, the fly actually develops as both male and female, which is very, very bizarre. But be that as it may, the important thing here is that this is regulated due to splicing. And so splicing in females leads to a female-specific double sex. No splicing in males leads to the male-specific double sex protein. RNA editing. RNA editing actually is not unlike what happens when you have selenocysteine. Again, it's a secondary structure which forms in your messenger RNA, but again, a specific secondary structure, not just a random secondary structure, although lots of those form. And that particular secondary structure is double-stranded RNA. That double-stranded RNA will be bound to by a specific protein. In this case, the adenosine deaminase active on double-stranded RNA. That's what ADAR stands for. And will modify this A, turn it into an inosine, and so it can base pair with other tRNAs and give you a different protein at that particular position. Translational control or post-transcriptional control, all about getting to the AUG. Can be some kind of repressor protein, can be some ribose switch, which is going to bind to some small molecule. My favorite are these thermometers, where just the heating up of the DNA, or say the RNA I should say, is going to give you access to the AUG, or sometimes you'll have antisense RNA. We talked about translational initiation. Translational initiation is completely dependent on getting that initiator tRNA to the AUG, and then hydrolysis of GTP when that happens. If you stop that GTP hydrolysis from happening by not allowing GDP to bind to your initiation factor, then you can block any kind of translation. Small RNAs, large RNAs do all kinds of really fun things inside the cell. Again, this should be kind of sounding like a broken record. It's a double-stranded secondary structure that forms in your RNA that associates with proteins, and those then lead to the regulatory aspects that are happening. The RNA sequence is serving as specificity. The proteins are what's doing the job. And so long non-coding RNAs are bigger than 200 nucleotides. The smaller RNAs are less than 200 nucleotides. These really small RNAs still are interacting with proteins. MicroRNAs are going to be RNAs which are made from genes that are present in the genome. Small interfering RNAs come from outside of the genome. They're not something which is specifically encoded. They both get chopped up into shorter pieces, about 20 or 22 nucleotides. This single strand of RNA here is associated with proteins. If it matches perfectly to an RNA, that RNA gets chopped up. If it matches partly to an RNA, translation is shut down of that particular RNA. Last thing that we talked about, which we talked about on Wednesday, again, could have spent a lot longer talking about it, CRISPRs. CRISPR is a wonderful tool for, I say CRISPR-Cas, I should say, is a wonderful tool for gene regulation and gene editing, but it was evolved to protect bacteria against viral infection. How does that work? The cell which is being infected by a virus somehow has to discover that this piece of DNA coming in here is foreign DNA. It does that through the PAM sequence, which is right next to the piece of DNA that gets chopped up. That piece of DNA then gets put into a CRISPR locus, which is the clustered regularly interspersed palindromic repeats, in the spacers, the colored ones here, relative to the orange ones, which are all the repeats. That CRISPR locus will get transcribed, giving you a big pre-CRISPR RNA. These get chopped into smaller pieces. This small piece now, the red one, can match a new virus which is coming in and chop that up. It's that process of matching the DNA to an RNA which is present in this CRISPR interference process. This is what people use for gene editing. It's that chopping up of the DNA relative to the RNA which is base paired with it. And that's why Jennifer Doudna and Emmanuel Charpentier will probably get the Nobel Prize before too long here. Not surprisingly, because this is a bacterial system which is providing resistance against viruses, resistance is futile. So you end up with an anti-CRISPR system, and almost all viruses, or many, many viruses, have worked out ways to deal with these kinds of CRISPR systems. Good luck on Tuesday. There will be extra office hours at 11 on Monday over in my office, and office hours now as well. Good luck on Tuesday. And then please remember to fill out those recommendation forms, or the evaluation forms. It doesn't have to be a recommendation, sorry. Evaluation.